I'm David Chen, the WuXi Advanced Therapy CEO. It's my great pleasure and honor to host and modulating the panel discussion session, Realizing the Promise of the Gene Therapy in Pompe Disease. It's my great honor to introducing three distinguished panelists to join this panel discussion. Dr. Barry Byrne, Associate Chair at the Pediatric and the Director of the PAL Gene Therapy Center at the University of Florida, and also the founder of Avanti Bio. The second panelist, Dr. Edward Connor, is Astellas Gene Therapy Site Head and the Division Head of Gene Therapy Medical and Development. And the third panelist is Jeff McKay, is a founder, president, and the CEO of Avero Bio. Let me get into the discussion right away. Maybe my first question I can direct to Barry. So Barry, what has been a main limitation of the enzyme replacement therapy, either the first and the second generation? Also, in your opinion, why is a gene therapy ideally suitable for the treatment of Pompe disease?
Well, thanks, David. I'm glad to be here and really enjoy visiting this afternoon with both Jeff and Ed. So I think this will be a lively and informative discussion. We began work in this area almost 20 years ago because of the unique circumstances of early onset Pompe disease that I began to see as a cardiologist, where the presentation of these patients was so challenging to manage. One of the most severe forms of early onset cardiomyopathy that we see in clinical practice. and certainly enzyme replacement therapy has been transformative for that particular aspect of the disease, because the exposure of recombinant proteins to the vasculature and to the heart assures reasonable amount of the protein is delivered into cardiomyocytes. What we began to see, though, in both patient populations and in the models that we had established of Pompe, is that there are manifestations that are not easily addressed through peripheral administration of recombinant proteins, because as a 110 kilodalton protein, this is not transported across the blood-brain barrier, and the same cellular effects of glycogen accumulation in the musculature, including the heart, are seen within neurons, and so those impact the neuronal function and ultimately their neuronal survival. And even in the long-term treated population, we see many manifestations of this in terms of facial weakness, respiratory insufficiency, inability to sit independently and walk in some of those patients. So that's what encouraged us to continue to pursue additional treatments to meet that unmet need in the patient population using this now technology that is become more pervasive in the variety of conditions that are genetically defined. So that's what we're focusing on in particular in the work we've been doing.
Great. But do you think that gene therapy can really addressing the fundamental root cause of the Pompe disease?
Yeah, conveniently, the alpha glucosidase gene is entirely incorporated into the AAV vector in a way that various control elements can be used to facilitate expression in a variety of tissue beds. So obviously there's the potential to have a version of enzyme replacement therapy where continuous expression from the hepatic depot of expressing cells is one option. Or what is, at least nature told us, is most efficient is to have the delivery and processing of the protein from a cell autonomous route. So that requires that the vector genome enter each cell that may need to have a correction of alpha glucosidase activity. And in that way, very low amounts are actually needed because the whole system was evolved to very efficiently traffic the protein from the Golgi to the lysosome. So.
Great, thank you, Barry. Thank you so much. Maybe I can direct a similar question to Ed. So Ed, so what might be gene therapy address the limitation on the ERT, those current traditional treatment?
Yeah, I would echo a lot of what Barry said. With regards to ERT, it really has been transformational for the lives of patients, not just for Pompe, but for a whole host of rare diseases. I think one of the challenges, as Barry pointed out, is its ability to get into the cell. And so typically what's done is to give large amounts in the hope that you're creating gradients, because the receptor that brings it into the cell is a relatively low affinity receptor. So just as Barry pointed out, the two options are to produce steady and consistent levels, whether that's through liver-directed gene therapy, whether it's through the approaches that AVROBIO is taking, or to direct it to the tissue of interest itself. And that's the approach that we're taking, is we're utilizing an AAV viral vector that we know well. We know which tissues it goes to. We know what the viral transduction, how it gets its genetic contents into the cell. And we have experience understanding how does the mouse model translate to the non-human primate or monkey model, and ultimately translate to humans itself. And so that's where I think gene therapy, as Barry said, can provide real advantages because it should be able to get more of the enzyme into the cells that need to express it.
Great. Maybe even go deeper for using the gene therapy to treat the Pompe disease. So which population of the Pompe disease patient were expecting have the bigger, better benefit? For example, the early versus late, intramuscular, central nervous system.
Yeah, it's a great question. I think it really depends on the construct and how you design it. Barry mentioned a lot of the central nervous system involvement that's present in the early onset patients. And so, some gene therapies or some viral vectors, for example, have the ability to cross the blood-brain barrier. Others do not. And then the promoters that you use to drive expression will also affect where you're expressing GAA. I think one of the limitations that everyone's aware of for gene therapy is understanding how those viral vectors, which tissues they get to, which cells they get to, and how that may ultimately affect what it is that you're able to improve with regards to the disease pathogenesis. So to answer your question, for example, we're looking at muscle-directed AAV, and we're starting with late onset Pompe disease patients in an effort, again, to drive expression within the muscle. I think one of the open questions is, will there need to be different constructs to address different aspects of the disease? And then how broadly can you have that expression occur, and how broadly can you transduce those cells, as Barry was talking about earlier, so that you can get either direct cellular expression, or again, you can have enough steady and consistent levels of the enzyme that it can ultimately have a stronger interaction with the receptor and be brought into the cells of interest.
Great, thank you so much. Any additional comment for what we're learning from Barry and Jeff, or anything to add?
No, I mean, I'm completely aligned with how they view this. Yeah.
Maybe I can shift the gear towards about how gene therapy being administered, right? Maybe Jeff, Can you share your thought about what are the key consideration or the challenge you have perceived in gene therapy to be used in treating the Pompe disease?
Yeah, happy to. So, we at Avro are developing lentiviral gene therapy for 5 or 6 different lysosomal disorders. And so when we look at Pompe, we can't help ourselves but to try to compare and contrast what is the same and what is different, just because of who we are and how we spend our day. But of course, starting with a target product profile to sort of clarify what we're trying to achieve on the most high level, like all of us, we're trying to provide this long-term, durable, maybe even lifetime response with a single infusion. But when we look at Pompe specifically, it to us is the most challenging of the lysosomal disorders that we're targeting. And again, comparing and contrasting, just starting with there are 3 challenges that I think have a higher bar with Pompe. First of all, we feel we need 20 to 40 times as much enzyme in the ERT world as a dose. So very high levels of systemic distribution relative to, for example, Fabry disease or Gaucher disease, some others that we focus on. So that's one hurdle. But the second thing, as has been mentioned, is we really need to impact the muscle, including pulmonary, respiratory, in a meaningful way. And then thirdly, and this, we've learned from Professor Byrne and others about how important the brain and the spinal cord is, and that this is a brain and spinal cord disease. So we need to have that global CNS effect, motor function, cognitive impairment. So that's the starting point, is that our lentiviral gene therapy, but with these extra hurdles. And in our preclinical models, what we demonstrated to ourselves, at least, was that our standard sort of off-the-shelf ex vivo lenti gene therapy met some of those hurdles, but not all of them. And so that caused us to think about what needed to be engineered or addressed differently. And we really have tried to focus on a 3-pronged solution, which is, number one, to generate that systemic expression in a prolific way, meaning higher levels of enzyme than our other disease targets, primarily via a more potent promoter, but some other approaches as well. And then number 2, to incorporate a lysosomal uptake tag, a GILT tag, that many would be aware of its early use in BioMarin, because we felt that this has already been shown to increase uptake in the muscle 25-fold. So this sort of 1-2 punch of lenti gene therapy plus the GILT tag, but also to combine our gene therapy with busulfan conditioning to facilitate engraftment of the corrected cells, not only in the bone marrow, but also in the microglia compartment. And so we felt with this, 3-pronged approach of, a more aggressive promoter, GILT tag, combining with busulfan conditioning, we could ideally, this is aspirational, all we've demonstrated is great efficacy in Pompe disease mice models, but that we would hope to be able to check those 3 boxes of systemic distribution that's more prolific, targeting muscle, targeting brain. So that's where we are now. We've completed our preclinical work and look forward to, testing it. Now, our first focus area will be classic infantile onset Pompe, and that was reflective in our preclinical studies. But of course, that's an area of very high unmet need. So we'll be interested to see whether or not the technology translates from preclinical, from animal to human.
Yeah, thank you. You probably know this question coming to you, right? AAV delivering vehicle has some limitation, and you're such experience and you know so much about the lenti. Do you expecting the lenti delivery can circumvent some of the AAV-based kind of gene delivery?
Well, I do, because I think for fair balance, they each have their strengths and they each have their weaknesses, and I think they're different, right? So, for fair balance, I don't, before I talk about some of the strengths of lenti, I think that the challenges primarily can be drilled down to it's a complex personalized therapy. We modify the patient's own stem cells outside the body and then reinfuse. So there's a complexity that doesn't exist with AAV. And then there's the tolerability of the conditioning approach, which we're working hard to minimize, but it's an aspect of the therapy. So those are our challenges, but I guess the challenges related to liver toxicity or immunogenicity are not seen to date in Lenti. And I think that some of the benefits of using this personalized approach, complex as it is we're not having to address things like neutralizing antibodies to the AAV itself, and even to the enzyme. I think that there's been early data in diseases like Hurler's and in our own Fabry program that the enzyme, that the procedure itself may tolerize the patient to the enzyme. And so, and I think in terms of directed uptake, we just have a completely different approach, for better or for worse, is that, the primary mechanism is that we transduce billions of leukocytes, which both manufacture and distribute protein. So some of that distribution, for example, a transduced macrophage would home to inflammation. And ideally, if our drug is working properly, these messengers will be carrying enzyme throughout the body, head to toe, and that's what we're aiming to address. And then, of course, there are manufacturing complexities on both sides for AAV and for Lenti. They just happen to be different. Is that in our world, a large bioreactor would be 200 liter, where we can manufacture in a single run enough to treat 40 to 50 patients in a 200 liter serum-free suspension bioreactor today. So, in the rare disease world, that should be enough to treat thousands of patients a year. And then we've invested very heavily in automated manufacturing. So now we're up and running on 3 continents to be able to manufacture in what is called the Miltenyi Prodigy system that we've customized for gene therapy. So none of that is easy, but I think that the manufacturing on both sides of that ledger still are a work in progress.
Fantastic. Any thought, Barry, about comparing AAV and the Lenti, the pro and con? Oh, Barry, I think you are silent, Barry.
Excuse me. So as Jeff mentioned, the fundamental difference is whether these genomes are stably incorporated into hematopoietic stem cells or episomally delivered directly to the target tissues, whether they're lower motor neurons or muscle cells, and there may be benefits from either approach. They is exactly as you said. They have strengths and weaknesses. In the focus on early onset Pompe and recognizing the fact that AAV vectors are resident episomally in the cells, a lot of our focus has been on dispelling a conventional wisdom that AAV delivery is a one-time delivery strategy. And so we've spent the last several years establishing the basis for readministering AAV vectors so that there can be sustained effects over the lifespan, because right now in the US, about half of children with Pompe are diagnosed as newborns. That will continue to increase over the coming years. And so we will then have to make a decision, even whether they're early onset or late onset diseases, when and how to intervene to try to optimize outcome. So the paradigm is really changing. Since we implemented newborn screening in Florida, where we have about 250,000 births a year, we've had identified 5 patients with Pompe disease, and all late onset. And so that's a very challenging discussion with the families, and they are the only comfort they derive from what they might tumble to thinking is a devastating situation for that newborn, is that there are options like these that are being discussed today available. And that in fact, they will hopefully have many options. So This is, there are some fundamental differences, but in the end of the day, the goal is to try to provide the best, most effective therapy for all patients, young and old, and not just the existing patient population, but those that are being identified really early, where there's probably a even better chance to have a long-term impact.
Any additional thought, Ed, from what you heard from Barry and Jeff?
Yeah, I would echo the comments of Barry. I mean, I think, there used to be a pretty clear distinction between gene editing and gene therapy approaches. And retreatment was one of the things that was, seen for a long time as, very distinct between those two. And I think those lines are blurring. I think what Barry's group has done has been phenomenal. There's been a lot of research, generally speaking, understanding from an immunologic perspective, how can we potentially retreat, or even with both of these approaches, there may be immune aspects of initial administration that we aren't yet aware of. But I think that's something that the field is learning very rapidly. And so I would also echo Barry's sentiment. From my perspective, the most exciting thing is that so many companies are working on treatments and utilizing a lot of different approaches to do that. I hope that they're all successful, and I know that even those that aren't successful, we're going to learn, and it's going to help advance the science of the disease.
Yeah.
Thank you so much, Ed. Yeah, maybe Barry, along the lines of thought you brought up about the timing of the treatment, right? So do you think there's a optimal timing for the gene therapy to be administered is really most efficacious for the Pompe disease?
Well, we do know from the experience that early interventions will matter. That experience is growing in the U.S. as well, although we don't have as long term follow-up but the opportunity to identify patients early will help the community of providers establish better natural history data, identify other both imaging and molecular biomarkers of disease severity that can be a guide to when there are critical changes. Make an analogy to some of the work that's been done by the group at our institution to understand natural history in Duchenne muscular dystrophy by utilization of an MRI imaging biomarker. So the Imaging Neuromuscular Disease group and has incorporated some of those ideas into work being done by Jordi Diaz in the Pompe community. And so similar observations that there may be some predictive biomarkers that would be used to guide treatment.
Maybe I would another question directed to Ed, right? So what do you think the current challenging of the gene therapy in the Pompe disease from your the drug development side, that point of view? Any challenge you can share with us?
Yeah, I think the challenges are on one hand a good one. I think with the data that we've seen coming out recently at the World LDN meeting, that the bar is being moved for what available therapies are able to accomplish. And at least for where we are now, and this will obviously be solvable, most of the patients who will be treated with a background of enzyme replacement therapy, and whether that's traditional enzyme replacement therapy or as it develops remains to be seen. And so I think going then to what Barry said regarding endpoint development, it can be difficult to discern the effects of your gene therapy with an overlay of a patient who's already on enzyme replacement therapy. And so I think a lot of the studies you'll see will have randomized withdrawal of patients or withdrawal of patients if it appears to be safe. And again, that just means that the amount of time it takes to determine if an approach is successful or not might be somewhat delayed. I think the other way of looking at that question is also with regards to the DMD community and the MRI measurements. I remember being on a panel pre-COVID for hemophilia and someone asking why so many companies are doing hemophilia gene therapy. And the answer is because from a drug development standpoint, to have a blood biomarker that's tied, so one-to-one with bleeding risk and with the need for, with the need for therapy, that it's very easy is the wrong word, but you can determine whether you're having an effect or not relatively early in the course after you've administered the drug. And I think that's where I give a lot of credit to the DMD patient advocacy community. I think they've done a lot of that work, understanding what are the risk factors and what the risk factors, how that predicts how endpoints in terms of muscle strength will progress over time. With Pompe, there are of course the standard assessments that we look at, like 6-minute walk, like forced vital capacity in an adult patient or in an adult patient group, for example. But I think that's where the field will head, is that we'll have both objective as well as patient-reported outcome measures that really do help us discern, are these drugs working or not? Because, as was said before, so many people are working in the field, which is great. And we need to, in a systematic way, to be able to determine, okay, this works, let's move forward with this, or this doesn't work, so let's try something else. I think there's a lot of lessons that can be learned, for example, from the field of oncology, where they look at development in a very rational way. Now, again, they are able to, do scans and look at how much the tumor has grown or not grown. But I think with regard to radiographic measurements, I think with regards to biomarkers, whether it's in the blood or the urine, the field is progressing, where you will be able to get an earlier readout about these therapies and whether or not they are successful.
Any additional comment, Barry, Jeff?
Yeah, I think despite the pandemic, we were able to complete through the Muscular Dystrophy Association the patient-focused drug development activity that has led to a voice of the patient statement that really should be guidance for everyone developing drugs in the Pompe patient community. And one thing that comes across loud and clear from patients is that existing therapies still leave some room for improvement because of the fatigue that patients face. And this is debilitating for many individuals, because it's hard to really live your life under those circumstances. So that should be part of our guidance as to whether these are detailed Pompe specific quality of life measures or focused on a fatigue rating scale. We may be able to find that alternative therapies actually impact that aspect, and one would expect it to, in fact, because when you look at through the Pompe registry, we actually over many years and about 1,600 patients assessed rate of change in some key domains, and on treatment, there is still a negative trajectory in some key outcome measures. So if an alternative advanced therapy, whether it's lentiviral mediated gene therapy or LOPD, muscle-directed gene therapy, or systemic gene therapy in IOPD, you would hope to see a positive change in some of these key outcome measures and not just a reduction in the rate of decline in patients. And I think that's what the patient community is now expecting and should expect from a successful treatment strategy.
Fantastic. Any additional thought to add, Jeff?
I mean, I think that encapsulates it. Maybe just to agree, I mean, one thing that we found interesting in this field is, when we've done focus groups and research to try to really understand the needs, if you have two columns, what the physicians characterize as the needs in ranked order, and the patients, the clinicians are often focused on end organ damage and others. So we really do need to, target and measure the quality of life aspects.
Thank you. I think with the interest of time, I probably concluding with this the final question, right? I think we have seen a lot of increasing progress in collaboration among the drug developer, patient, and doctor to advance the treatment, especially the gene therapy to treating Pompe disease, right? So from the drug product perspective, right? Do you see any major gap that still need to be addressed now or in the next decade? Maybe I can direct this question first to Jeff. Would you mind to comment on that?
Well, so I think it's one of the challenges in designing trials is that you have to sort of placate many masters, and the FDA is the one that probably is the least flexible in terms of meeting those needs. So, I think we're always trying to understand, first of all, what is the primary endpoint that is regulatory mandated, but beyond that, we'll have greater flexibility in the secondary endpoints or the exploratory endpoints, where we can do a much better job in really connecting with the patient community and understanding what are the things that are really important to the community to properly measure.
Any thought add on the same question?
Yeah, I would very much echo Jeff's thoughts. I think one of the things that we tend to approach drug development in the way that we've always known it for Pompe, but I think one of the things that we need to be open to is, as these treatments roll out and as patients hopefully respond to them, I think thinking about the way that we look at responses. I think the fatigue response, the fatigue example is a great one, but I think these have the potential to be transformative therapies, and so understanding and picking up on those signals early, I think, and sharing that information, I think, will be very helpful, because oftentimes the scales are adjusted to, in some cases, measure incremental benefit. And if you're seeing more than just incremental benefit, then you need a scale to be able to understand the magnitude of that effect. And so, I think it's upon all of us to make sure that we're monitoring that, and then coming back to the scientific community saying, hey, maybe there is a better way to measure this.
Barry?
Well, one part of my life is in the more basic science aspects. So we've tried to facilitate expediting the drug development timeline through establishing non-clinical models that I think are an aspect of the pre-competitive space that maybe you're alluding to, where we can share, both to the scientific community, trying to understand mechanism, and to drug developers to help de-risk clinical studies, so they're not iterative in humans, but rather you have put your best foot forward when you responsibly use representative animal models. So we've, in our initial collaboration with Nina Rabin, the full knockout model, we've established 2 other conditional knockouts, as well as 2 rat models that recapitulate the early and late onset disease, as well as the naturally occurring canine model, which has been informative, and by understanding its natural history. So, that if that helps the overall scientific community and helps the patient community, then I feel like we've done our part to help expedite the drug development path for this area. So that's been rewarding.
How would you see the gene therapy evolve for Pompe disease treatment, right, in your estimation for the next decade or so? What's your crystal ball tell you? Maybe Jeff, what's your thought?
I mean, I think the great situation that we're in is that there are multiple approaches. So, you know, we don't need everything to work. We need something to work. And after years and decades of research, they're all either in the clinic or approaching the clinic. So, you know, I think we're going to do an awful lot of learning in the next handful of years about, you know, the pros and cons of the various approaches. And, you know, it might be that it's not a one size fits all, that there's different segments of the population that are better suited for various approaches. But I think the exciting thing is there's just so many energy units being applied to this very important indication that we just have to hope that, you know, it translates. But it's been a long wait, and hopefully we'll be getting some pretty exciting answers very soon.
Great. And Ed, any thought?
Yeah, I would. That resonated a lot with me. I mean, I think it's at conferences like this, sharing the information, making sure that everyone is, you know, as Barry was talking about earlier, you know, making sure that we're doing as much information sharing as possible. And, you know, as was said, there may not be a one size fits all approach, but again, learning from that and then iterating upon it, I think is going to be very helpful.
Barry?
So you asked for the crystal ball. I wish I had one, but I do think that providing options to patients is going to elevate their expectations and hope of improved quality of life. And but it is challenging in an era where we have to focus first on safety. This for better, worse does slow the process. And there's new guidance now from the agency on guidelines for in neuromuscular diseases, and there are important improvements anticipated in the critical quality attributes of the gene therapy agents that are being used in many of these programs across the disease spectrum. So you know, it will be incumbent then on industry where the therapies have to come from to be durable, right? We need our industry partners to help develop these drugs in a way that they'll be available for all time in the future to new patients identified with Pompe. So we just have to meet that standard and make sure that we have safety first and that we reliably demonstrate that they're effective and durable.
Thank you, Barry. I really interest of time. Any final thought? Ed, Barry, Jeff? Jeff, maybe.
I mean, really, just a quick thank you. It's a real treat to actually get to talk about this issue with some of the real leaders in the field. So thank you.
Thank you, Jeff. Ed?
Yeah, again, thank you. It was an honor to speak on this panel. Really, really enjoyed it. And thank you very much for the opportunity.
Yeah, thank you, David. Thank you, David. Thank you, WuXi. And we hope this is informative to the people who participate and see and learn about from this in the future. So we appreciate it.
First of all, thank you for being participating in this panel discussion. I personally have learned a lot. I'm sure the audience will just learn as much as I do and feel exciting, not only for the promising current status, but even encouraging for the future. I thank you for your great work you have done for the Pompe Society, right? I know those unmet medical need patients are waiting for us. I will be cheering for you. I will try to contribute from my part to making this breakthrough therapy, either using traditional enzyme replacement therapy or the gene therapy that's dear in my heart to work eventually with transforming the patients, their current life. So thank you so much for your participation. Thank you.